Hello everyone and welcome to my video. I'm Dr. Mahdi. Today and in this video I'll explain to you about different protein structures. I mean primary, secondary, tertiary and quaternary. At the end of this video, if you like it, please push the like, press the subscribe button and also share it with your friends. Now let's start the video. I would like to start with primary structure. Protein is a polymer and monomer for this polymer is called amino acid. There are 20 different types of amino acids. In the structure of every amino acid, in one side there is amine group or NH2 and other side there is carboxylic acid group or COOH, but R should be different in different amino acids. Depending on R, they will be classified to basic, acidic and neutral. Two amino acids can be connected to each other using peptide bond. Peptide bond is a reaction between amine and carboxylic acid group, and one water molecule will be released. The final product is called dipeptide. I mean, we have dipeptide when two amino acids are connected to each other using peptide bond. But when we have between 3 to 10 amino acids, between 3 to 10 amino acids are connected to each other using peptide bond, the final product is called oligopeptide. And if a number of amino acids is more than 10 amino acids, it's more than 10, definitely the final product is called polypeptide. Here you can see different amino acids are connected to each other to make a polypeptide. And now secondary structure, depending on hydrogen bonding, there are two different types of secondary structure. I mean alpha helix and beta sheet. Alpha helix is because of hydrogen bonding between CO and NH in two different amino acids in one strand. I mean here we have a strand and here you can see hydrogen bonding between CO and NH. All of them are hydrogen bonding, and after that strand will be a spiral, and in every turn there are 3.6 amino acids. And here you can see alpha helix structure. I mean after hydrogen bonding, a strand will be spiral. The second type of secondary structure is called the beta sheet, and again beta sheet is because of hydrogen bonding between again CO and NH, but in two different amino acids in two strands. Alpha helix was because of hydrogen bonding between two amino acids in one strand, but beta sheet is because of hydrogen bonding between CO and NH in two amino acids in two strands. And there are two different types of beta sheet. I mean parallel and antiparallel. What's difference between parallel and antiparallel? As I already mentioned in the primary structures, we have a polypeptide. I mean different amino acids will be connected to each other using peptide bond, and we will have a polypeptide. Definitely for every strands of polypeptide, in one side there is a carboxylic acid group and we call this side C terminal, and other side there is amine group NH2 and we call it N terminal. Suppose we have two strands with a C terminal and N terminal. If C terminals and R terminals, they are close to each other, we call it parallel. But if they are far from each other, for example, here we have C terminal and other side again C terminal, N terminal, N terminal. If they are far from each other, we call it antiparallel. And in antiparallel, C terminal is close to N terminal. But in parallel, C terminal is close to C terminal. And antiparallel for beta sheet, antiparallel beta sheet is more stable than parallel beta sheet, and it's because of nice alignment of hydrogen bonding in antiparallel beta sheet. And also here you can see beta sheet structure, I mean hydrogen bonding between two different strands. And also please note two strands can be same or different. And now tertiary structure. Before tertiary structure, protein is two-dimensional, but after that it will be three-dimensional. Different parameters, 4 different parameters help to bend and twist the protein and make it three-dimensional. And I would like to start with hydrophobic interactions. Hydrophobic interactions is because of interactions between alkyl groups in different amino acids. You can find alkyl group in, for example, alanine, it's alkyl group, or valine. And after interaction between alkyl groups, we will have hydrophobic interaction. Hydrophobic interactions because of alkyl groups in different types of amino acids. The next one is disulfide bridges. It's the most important one. Disulfide bridges is because of oxidation of thiol groups. For this purpose, we need an amino acid with thiol group, and you can use cysteine. When you have cysteine in the structure of your protein, definitely you will have thiol, and after oxidation you will have disulfide bridges. Next one is because of hydrogen bondings between different side chains, and the last one is ionic interactions. It's a reaction between different ions in the structure of proteins. For example, some parts of protein they have negative charge, some part they have positive charge, and after that we will have ionic interactions. And after tertiary structure, protein will be three-dimensional, and it will have a structure like this. You can see it's three-dimensional. And now quaternary structure, you cannot have quaternary structure for every protein. If you would like to have quaternary structure, you need to have at least 2 different types of polypeptide. When you have only one type of polypeptide, you cannot have quaternary structure. But anyway, for every proteins, we have multiple polypeptide chains, protein subunits. Sometimes we have same subunit and we call it homodimer, and sometimes we have different subunit and we call it heterodimer. But anywhere after quaternary structure, protein will be larger, and also for making quaternary structure, we need different parameters such as hydrogen bonding or disulfide bridges. And after that, we will have something like this. Again, it's three-dimensional, but in this case, it will be larger than, tertiary structure. Hope you found this video useful. At the end, if you have any comment or question, you can write it below in comment section. If you like it, please push the like, press the subscribe button, and also share it with your friends. And at the end, please don't forget that life is short and enjoy it. Thank you so much, and bye.